Hi there. So I'm an assistant professor at UW Green Bay, like Dr. Brusich, and in my lab I actually study ovarian cancer with some colleagues and undergraduates. But that's not what I'm going to tell you about today. What I'm going to tell you about today is something that I teach in one of my classes, human genetics, and that's about CRISPR genome editing. And so this is a relatively new technology within the last 10 years, but it has revolutionized both research and it shows promise for revolutionizing medicine and agriculture as well. Okay, so here's an outline for what my talk's going to be about today. First, I'm going to give you a little background about genetics, just to get us all on the same page, some keywords that you're going to want to keep an eye on for my talk. Then I'm going to tell you a little history about where the inspiration for CRISPR came from, and then what the technology is. So I'm going to make you wait a little while to get that information. Then I'm going to tell you some applications. What can we do with this technology? How are we using it? Not me personally, but how is society using it, researchers in medicine? And then finally, I want to start the conversation about when should we use CRISPR and when should maybe we have some restraint? Is this okay? Can people see me and hear me okay?
I think it might be a little hard to hear. But,
okay.
If you can speak a little louder, or...
I will just take it off. All right, so in all of our living cells, we have 4 biomolecules. So those biomolecules are the lipids, the carbohydrates, the proteins that are the workhorses. They're actually the ones doing a lot of the jobs. And then we have our nucleic acids. Those are the DNA and RNA. So I like to have a little model. That's the best thing about being a professor, is you get to have nerdy tools like this. So there's a model that I share with another professor, Dr. Pott. Don't tell him I took it, because if I break it, he's going to kill me. But when that information, so in every one of the cells of an individual, DNA is present, and that encodes the instructions for how to make that individual and to maintain it. Okay? So we call these blueprints, these instructions, we call these the genome. Okay? And the genome, again, is present in nearly every cell of that individual. Okay? This genetic information, this genome, is passed on during cell division from cell to cell, and it's also passed on through reproduction, so it's inherited. So I'm going to go to my model here and talk a little bit about DNA. So genomic DNA in the cell actually exists as a double helix. So you can kind of see it like that. So it's double-stranded. There are 2 strands of DNA that interact with each other end to end. Okay, so it's head to tail is the interaction. So what is also we can observe with the DNA double helix is that what is holding those 2 strands together. So what's holding those 2 strands together are the bases. Okay? So they're complementary to each other. So there's DNA sequences on one strand, A's, C's, T's, and G's. Right? So a different sequence on one strand. The other strand complements with it. Okay? So the other sequence of A's, T's, C's, and G's, and they pair with each other. So maybe you heard about this before, that A pairs with T and G pairs with C. So if there's an A on one strand, there will be a T on the other. And if there's a G on one strand, there's a C on the other. And that's what helps hold the DNA double helix together. So that's going to be important for later. I want us to understand that we call that base pairing. Okay, so the formation of interactions between 2 strands that's mediated by that complementary base pairing. The last thing I want us to understand about our genetic information, our genome, is that there's segments of our genome that encode information on how to make RNA molecules and proteins. So we call those genes. Dr. Brusich mentioned those earlier. Okay, so that's just a segment of a chromosome that encodes the information of how to make that product. So as I mentioned before, nearly every single cell in our bodies have this genetic information, has an identical or near identical genome. So how do we have over 290 different cell types? Okay, so how does that make sense? We have the same genome, but somehow we have muscle cells, we have eye cells, we have skin cells. Right? So how that turns out is that not every single gene is expressed in every single cell type. Right? And how I like to refer to that is some genes are turned on in a cell type and some genes are turned off. Okay? Collectively, we call that process gene expression. So expressing the gene means to make the RNA product and then make the protein. So how that happens is through gene expression. How gene expression happens is that first, whoa, hi, make that go away.
Just touch away from it.
Nope. There we go. Sorry.
It's a touch
I
screen.
can't touch these things. All right, so how that works, how does gene expression work? So first, a gene, right, a segment of DNA, first gets copied by some machinery in the cell into an RNA molecule. Okay? So it gets copied. We call that transcription. Then that RNA molecule is used as instructions for the cell to make proteins. So we call that process translation. All right, so the final product I want to point out, because this is going to come, and we're going to need to know this information later, is that sometimes the final product that's important is an RNA molecule, so this single strand of nucleic acid, and sometimes that final product is a protein. So I'm going to kind of ignore the carbohydrates and the lipids today. Okay, so sometimes researchers or even individuals might want to change the sequence of a genome. And we've had techniques to do that for quite a while, a couple decades, except the problem with all the other techniques is that they're inefficient and they are slow and they're expensive. So we haven't really had really good tools to be able to make changes to the genome. However, this new technology, CRISPR, has enabled fast, efficient, and relatively cheap way to make a change in a DNA sequence. So before I tell you about what that is, before I tell you how CRISPR works, I want to give you a little bit of history about how they discovered that they could make CRISPR, make this technology. Okay? So CRISPR is actually based on an intrinsic property of bacteria. So back in around 86 or 87, a paper was published where the researchers were studying the sequence of a bacterial genome, and they noticed something kind of interesting. What they saw was that there was these sequence that stop touching things. Sorry. What they noticed was that there is this sequence that is short. It's palindromic, right? So a palindrome is it's the same forward and backwards, like race car or my favorite taco cat. It's the best palindrome. So it was short, it's palindromic, and it repeated multiple times. So they called this clustered, regularly interspaced, short palindromic repeats. Okay? So when they were just analyzing the genome of the bacteria, they discovered these repeats. Probably 5 to 10 years later, they were still trying to understand what that sequence was, and finally what they found was that when they analyzed the spaces in between that are unique, they discovered that those sequences corresponded to viruses that infect bacteria. Isn't that interesting? There are viruses that can infect bacteria. We call those bacterial phages. Okay? So the non-repeated parts that are in between those repeats, those CRISPR repeats, are unique, and they correspond to other organisms. Well, viruses aren't organisms, but viruses. Okay? What they also found was there is a cluster of genes nearby that they called the CRISPR-associated genes. Okay, so that's kind of the background of what they discovered. How does this work? Okay, so what it turns out after studying what these sequences mean, what they do, is that this is the bacterial immune system. And so how it works is that if a virus infects a bacteria and it happens to survive that infection, the Cas proteins that were encoded by these Cas genes, some of them, help take some of that viral genome and insert it in between those CRISPR repeats. Okay? So it keeps some of its infection, that genetic information, and then what happens is that the genetic information, that genetic information gets transcribed, gets copied into an RNA molecule, and then it gets processed into an RNA called a CRISPR RNA. In addition to that, one of those Cas genes also gets expressed, and it forms a complex. That CRISPR RNA binds to the Cas protein. Sorry, I should just not touch it. That's even better. So the CRISPR RNA and the Cas protein come together, form a complex, and they look through the cell looking for viral genetic information. Okay? And how it knows when there's a viral genetic information is that the CRRNA will base pair with that invading virus. Okay? So it has to be a virus that has previously infected it, okay? Because that bacterial cell is remembering that it had been infected. And then when it makes a complex, when I am not good at this, my apologies. So when it makes that complex between the CRRNA and the viral genome, that Cas protein will cut it. So it's like molecular scissors. And pretty much that blocks the infection of that virus. So bacteria do this innately. Around 2012, 2013, about 3 or 4 publications came out that used this information and harnessed it to create this technology that can incorporate precise changes into genomes. Okay? So this was the inspiration for that CRISPR genome editing technology. All right, so now we can finally talk about how CRISPR works. So there's 2 parts to CRISPR that users need to deliver into a cell. The first thing they need is the Cas protein. Okay, so specifically it's called Cas9 because that particular Cas protein is small and it makes it easy to deliver into cells. Okay, so that's why it's called Cas9. It's just a particular gene. So that has to be delivered into cells. The other thing that has to be delivered into cells is called the guide RNA. And so that's a user-designed RNA molecule that will correspond, that will base pair with the sequence that you want to edit in the genome. So we call that the guide RNA. So you take genes that encode the guide RNA and that Cas9 protein, and you deliver them into the target cell, the cell that you want to edit. Then the cell will make the Cas9 protein, and it will make the guide RNA, so it will make the RNA molecule, not just the gene. They will form a complex, and they will find that place in the genome. It's guided, right, by that guide RNA. It's going to form that complex, and then when it forms that complex, the Cas9 protein will cut at that place in the genome. So we call this protein mediated because that's the scissors, and it's RNA guided because the RNA molecule is telling the Cas9 protein where to cut. All right. So where we've left off so far is that the Cas9 and the guide RNA come together, find the target place in the genome that is designed to be edited, and it cuts there. Then what happens? So they let the cell take over at that point. And in our cells, they don't like broken DNA, right? It's broken both strands. Oh, I'm not going to break it because Dr. Pot would kill me, but they break both strands, and the cell doesn't want broken DNA. So it's going to try to repair it, and it will repair it in one of 2 ways. One way is through random repair. Okay, so it will just join 2 ends of DNA together. It will take some bases off, it will add some bases on randomly, and it will fuse the DNA back into those single strands in the double helix. So this will change the genome at the location that they want, but it's not necessarily what a researcher wants. Okay? So sometimes this is all the researcher wants, or the user. But other times they want to make a precise change. They want to change just a single base from an A to a T. Okay? So in that case, if you want to make a specific change in the genome, you have to deliver one more thing to that cell. You have to give what's called donor DNA. And so that's the sequence that the cell should use to repair that break. So this is a system that takes advantage of the cell as well, right? Some of the cell machinery. Okay, so now that I've given you a brief intro to what the CRISPR technology is, what can we do with it? So with CRISPR technology, it's widely used in research at this point. Anytime anyone wants to study the function of a gene, they use CRISPR to alter that gene, and then you can ask the question, what happens? You can also make animal models to study human genetic diseases by altering the genome of that animal, and now you can study Duchenne muscular dystrophy or any other genetic disease that you would like cystic fibrosis. So this is widely used in research.
The middle button is a laser pointer too.
Oh, okay, thank you. Okay, so we can use CRISPR in agriculture. Well, not me personally, but it is an application for agriculture. So this can help solve some of the problems in our society right now due to growing populations and climate change that cause shortage of food. So they can make plants that are resistant to diseases. They can make plants that can grow in uncharacteristic conditions. They can also make food last longer by altering some of the genes to make them more shelf stable. They are even working on making gluten-free wheat and decaffeinated coffee. Right? So I saw some of those examples of more less important. They're still important, but less like large-scale important applications. So that's another application that has more impact on society. What about in medicine? So CRISPR is showing promise to have many applications in therapies and treatments and diagnostics in medicine. So there are trials and studies going on trying to help genetic diseases like cystic fibrosis, sickle cell anemia, which is a blood disorder, even cancer. They're trying to help our immune cells detect cancer cells better using CRISPR. So why I have this figure here is that I want to model two different ways that you could imagine using CRISPR in an individual to treat them for a disease. So some cells are amenable to being removed from the body, altered using CRISPR, and then delivered back. So the cells that give rise to blood cells, you can imagine, can be removed, altered, and delivered back to the patient. So that can get around having to use bone marrow donors, which can have its own problems with matches and efficacy. So we can imagine that that's going to be a really important application. Another application would be in vivo. So some cells you can't remove from the body, so those would have to be treated internally. Okay, so what are some limitations? The first limitation of using CRISPR in medicine is delivery. You can't get the CRISPR system, the Cas9 gene and the guide RNA gene, into every single cell in the body. Okay, so you have to be really targeted on what cells you can and can't treat. They usually use viruses, interestingly enough, to deliver the CRISPR system into cells. Another problem with CRISPR right now is there's a potential for immune response because it's a bacterial gene, so that might cause the body to reject it. A really big problem in CRISPR right now is off-target effects. So off-target effects can cause unintended changes to the genome that we didn't predict. That can cause more genetic diseases that would be bad. And finally, when should we use CRISPR and when should we not? So it's generally accepted that using CRISPR on somatic cells, so those are cells that don't give rise to new individuals in our body, like our limbs, our limb cells, our skin cells, our muscle cells. Okay? Those are generally accepted to use for CRISPR. Where the issue comes in is editing germline cells. And those germline cells give rise to new individuals. And so it's actually already been done. In 2018, it was reported by a group in China that they edited the genome of two embryos that gave rise to two little girls. Their pseudonyms are Lulu and Nana. And they had an edit to make them resistant to HIV infection. So their mother was HIV negative, their father was HIV positive. The goal was to make them unable to be infected from their father. So at the surface, maybe that seems great. However, the researchers did not make sure that the edit was safe. They couldn't prove that those girls were going to be fine, so they don't know long-term how that's going to affect them. That's also going to affect every individual that comes from those little girls. So they're making decisions not just for them, but for the future. So the result of this is that it was widely condemned by the scientific community, and it's currently, at least in the US, you cannot edit germline cells that will go on to become new people. But when the technology is safe, and we understand it, where should we draw the line for editing germline cells? right? Because you can imagine if someone has this debilitating or deadly genetic disease and they're going to die when they're 3 years old, wouldn't you want to be able to save them? Right? So one option for editing genome cells is you just can't. We're not going to edit cells that will go on to give new people. They can't consent to it. Another option is only edit genes that give rise to individuals who have lethal diseases. They will die very early. Alternatively, you could have just debilitating genetic diseases, right, that cause them to have a standard of living that would harm them, right? And then finally, what if we have no restrictions? What if we want to edit whatever we want? So that's where I'm going to leave us, and we can start the conversation.
Oh my God. Woo! Thank you so much, Dr. Heyrman. If we had some special effects right now, I'd request that emoji with the like exploding head, that was like print over my face right now, because I mean, really testing the plasticity of my mind to take this all in. Thank you so much. So we have some really thoughtful questions. How accessible is CRISPR? Is it really expensive?
No, it's actually like I have a little bit of funding. I could probably make it work in my lab. So I can buy the resources. I can then amplify them in my lab and do it myself. So I haven't yet. I plan to eventually in my ovarian cancer cell line. So I don't work in people, but I could use it for my own work.
So that kind of gets to one of the other questions. Trying to imagine what the mechanism is for performing CRISPR alterations.
In people or in
people or in just cells. So say, like you were saying, you could like, even for people, extract cells and what's the correct way to say? Right,
yeah.
Edit them using CRISPR technology. What does that look like? Like at what size even, trying to imagine?
So at least if you're going to take them out, you can culture cells in kind of petri dishes, or we use flasks. You have to use very safe conditions so that you don't contaminate your sample or contaminate a person, right? So it's actually very microscopic, just like this picture. If they were working on just cells, it's just as simple as that.
This is what that would look like.
They would work in biological safety cabinets, though, if it was on cells of higher organisms, not just yeast or something.
Thank you. How to, and this might not be the right term, but you mentioned something like some of the risks, was it off-target mutations?
Yep.
Okay, so how to test against that if we're putting it in, say, food? You know, I've heard discussions around
absolutely
the safety around that.
That is a whole nother discussion I didn't even talk about, is making sure that we're making correct edits that are going to end up in our food chain. And so every single change that you're going to make to an organism has to be thoroughly tested for safety before it can actually make it to market. Because it is a really real risk that you're trying to edit this part of the genome and unfortunately a different edit happens. And so they are working on the technology to make it safer and make it more precise.
So how many times does something need to be tested? Is it like into the thousands or millions, or?
I am a basic researcher, so I actually I don't know that answer. No, that is a great question, and I'm sure that people who work in that field, that it is thoroughly tested before it can be in the market. And even there are some examples where something unexpected happened that actually gets pulled from the market.
Oh.
Before, if something is shown, they will take it from the market.
And then what led you to delving in more deeply, you know, with your ovarian cancer research into CRISPR?
So actually, I don't use CRISPR yet. I just think it's a really powerful tool for my research. So I'm actually not an expert in CRISPR. Right? This is something that it's interesting to me, and I teach it, but I would like to use it as a tool more so than I want to study how CRISPR works.
Thank you.
Yeah.
Any more questions coming in? I saw the chat going.
Yeah, we have time for one more. Let's see. How are the viruses inserted into the bacteria?
So viruses, they're just very good at infecting cells, as we all know right now with COVID, with the virus, the coronavirus currently right now, is that's what they do. So viruses aren't living organisms; they're basically genetic material that is encapsulated in some protein, and it finds a cell that it can recognize, and it inserts its genetic material into that cell. And so there's specific viruses that infect bacteria.